Dr. Dwight Nisley, director del Programa de Tecnología de Investigación del Cáncer en el laboratorio Frederick National Laboratory for Cancer Research. Desarrolló parte de su carrera en la investigación de elementos móviles, retrotransposición, mecanismos de transcripción inversa, fidelidad del VIH1, transcriptasa inversa y la evolución de la resistencia a los medicamentos durante la infección por VIH1. Posteriormente utilizó su experiencia en biología experimental para supervisar y gestionar los esfuerzos de investigación básica del Centro para la Investigación del Cáncer del NCI. Realizó su doctorado en genética y biología molecular en la Universidad de Notre Dame en Estados Unidos. Les pedimos un gran aplauso y una calurosa bienvenida al Dr. Dwight Nisley.
Thank you all for coming today. Thank you to the organizers for the invitation, and thank you especially to the students. I think the highlight of this meeting has been to meet students and trainees and talk to them, to see your excitement and your passion for working on cancer and finding cures for this terrible disease. The city's beautiful, the venue's nice, and I've really enjoyed this meeting, so thank you for inviting us. Again, I'm Dwight Nisley. I'm at the Frederick National Lab for Cancer Research, and I run an entity called the Cancer Research Technology Program, which has multiple entities in it that focus on technologies that enable and carry out cancer research. One of those entities is the RAS Initiative, and I want to tell you a little bit about some of our work today in that area. Okay, when Harold Varmus and NCI leadership first set up the NCI RAS Initiative, the idea was that this would be an enabling hub or a nucleating effort that would go beyond the national lab. In the center of the screen, you see the building that we're in. But the idea was that we would interact with universities, the intramural labs, biotechs, pharma, and outside entities to try to nucleate new efforts to go after RAS-driven cancers. Tommy touched on this a little bit this morning, but I want to reiterate it. We work as a team. We're not an academic lab. We have many different groups, which are led by some of the people on the right, including Frank McCormick, who's our advisor, who comes to Frederick about once a month and spends a week there. But we have 70 people. They're all listed here. And they work in many different disciplines. We work on multiple projects, and we interchange and move rapidly to take on new projects and new challenges. Everyone's excited about curing RAS, and working collaborative has been a very strong component of our program. As Tommy walked over, just a little bit this morning, about 30% of cancers are caused by mutations in the RAS gene, particularly pancreas, colorectal, and lung cancers. Most of those cancers focus in a couple alleles or residues in RAS around G12, 13, and 61, which impact catalysis of GTP to GDP, which we'll go into a little bit more detail in a bit. And these are so-called oncogenic mutations that are associated with cancer. As Tommy again showed you this morning, there are multiple different alleles. One of the things that I'd like you to appreciate or to think about is that you can see that there are different alleles, and in different tissues, the prevalence of those alleles changes. So this is one of the arguments for directly targeting RAS, right, is that even though it's the same protein, the downstream effects, the way it engages effectors, and the multiple wirings that are involved are probably quite complex and tissue-specific. Okay, this is just a primer for those of you who have forgotten Bio 101, but it's just a good review of where RAS sits. So RAS is a binary switch. It's part of a pathway that transmits signal from outside of the cell to the inside of the cell and activates a transcription mechanism and drives proliferation. RAS is a switch. It is on in the GTP state, off in the GDP state. It's associated with the membrane. Its activity happens at the membrane. It's important for it to be there. And it interacts with effectors. In this case, we're just showing the canonical MAPK pathway, but it needs to physically interact with RAF in this case to transmit signal down that pathway. Okay, again, just a review of the transition from GDP, the so-called inactive or off state of RAS. There's a protein called SOS that helps exchange GDP to GTP, and it becomes active. Normally, in this active state, it'll engage one of the effectors like RAF or PI3K or RAL-GDS to drive a transcription program. And normally, another protein called a GAP, a GTPase activating protein, interacts with RAS and helps catalyze the conversion of GTP back to GDP. In the presence of oncogenic mutants, those at codons 12, 13, and 61, and some other codons, this process of actually cleaving GTP and converting it back to GDP is blocked or doesn't happen. So you have a constitutive stuck on RAS, and the signaling that goes through the pathway is much greater. This is just a cartoon that shows the mobility of regions known as switch 1 and switch 2, which are in different conformations when it's in its off state, GDP bound, or its on state, GTP bound. The reason this is important is that the GTP bound conformation of switch 1 and switch 2 are important for binding and engaging the effectors. Okay. Over the years, mutations in the MAPK pathway have been appreciated as drivers of many different cancers, and many attempts by pharma and biotechs to block this oncogenic signaling have resulted in a collection of drugs against many other components of the pathway that are in clinical use now. Some of them are effective, some of them are cytotoxic, some of them result in resistance through feedback activation of the pathway. The goal of our program is to go directly after RAS itself. Even though it's been studied for 40 years, there are still no clinically FDA-approved drugs for RAS-driven cancers that actually target RAS. So that's the goal of our program. And there are two major goals of the program, or two objectives, and that is to find small molecules that directly target RAS. Tommy told you this morning a lot about some of the cellular and the biochemical assays we've developed. I won't tell you any more about that, but there are many different type of assays we don't have time to describe today. There are also biophysical assays, which I'll go into a little bit more detail. We've used computational CAD approaches in silico screens to look for molecules that will bind RAS. And our basic philosophy is that we want to discover things, preclinical inhibitors or compounds, and then work with pharma or biotech partners to further develop those. In addition, again, Tommy described this a bit this morning, we want to better understand RAS-RAF signaling complexes in the context of the membrane. Most of the last 30, 40 years of RAS biology have been looking at RAS, the G domain, in solution. And we believe very strongly that understanding how it interacts with the membrane may reveal therapeutic opportunities that up until now have been underappreciated. Okay. This is a bit of an advertisement. I'll just run through this quickly. We've developed many capabilities within the RAS initiative. The reason I wanted to show them to you is that we love to collaborate, and we have capabilities that might be of interest to you. So these are areas that you could reach out to us and potentially work with us. We have a very strong biochemistry and biophysics group, all types of platforms for doing analysis that we can work with. We do a lot of structural biology. We've developed a lot of the assays. Tommy developed some of those described some of those to you. And both biochemical assays and cell-based assays for high-throughput screening, which we've used in partnership with some large pharmas. We're also looking to identify new targets, synthetic lethal interactions with RAS. We've set up a synthetic chemistry capability that leverages some of our biophysics and structural biology components. And we've also set up a new computational chemistry capability to do virtual screens that are based on docking and some MD analysis of ligand binding to specific sites. And then we have this very large RAS reagents program, with the idea here being is that the cell lines, the vectors, some of the protein reagents that we've developed, we want to make available to the world. This is what our hub and spoke looks like. We collaborate with a lot of academic entities. We have strategic collaborations with some of the national labs. We've signed CRADA's research development agreements with many pharmas to try to develop some of the compounds we're working on. Okay, and just an example of the reagents, Dom Esposito, who runs this program, is looking for world domination, and this is how far he's gotten in his attempt to get our reagents out across the world. I was happy to see that Mexico's already on the list. Okay, so current efforts to target RAS. This is just a quick review of some of the published papers that you might be aware of where people have attempted to find small molecule inhibitors that would abrogate RAS signaling. One of these is to look for compounds that bind to the SOS pocket. Remember, SOS is the activity that promotes the exchange of GDP to GTP. So the idea being if you can't load GTP, you can't become oncogenic. We don't favor this approach because most of the RAS mutants are stuck in the GTP state. Nevertheless, there are compounds that have been identified that bind to the so-called SOS pocket. Another approach that's been taken, and this was done primarily through a small biotech in San Francisco and Frank McCormick's lab at UCSF, is to use a SHG or second harmonic generation platform. And very quickly, this platform uses reflected light on a membrane that has a resonance that can be measured. You do this by placing a dye on your protein of interest. If a molecule binds to that protein, it will shift, potentially allosterically, the angle of that dye, and the resonance that comes off changes. And that allows you to screen large numbers of compounds. So several compounds have been identified that way. They've been confirmed by NMR analysis in the upper right, and there are small shallow pockets that they may bind to. The Rabbits lab has identified some compounds which they believe would prevent RAF, the RAS binding domain of RAF, from actually binding to the RAS protein. An example of that is shown right here. And those may have some promise. One of the things we're always looking for is specificity. KRAS is important in cancer. The related proteins NRAS and HRAS look very similar to KRAS. So blocking their activity or their ability to interact with RAF would potentially be quite cytotoxic. So we'd like to find things that are specific for KRAS itself. So some of these compounds will block those interactions, but they're probably not KRAS specific. Most of you are probably aware there's a new class of inhibitors referred to as covalent inhibitors. These take advantage of the fact that one of the oncogenic mutants, G12C, is a cysteine. Cysteine's reactive and you can derive covalent compounds that will attack that. G12C is prevalent in lung cancers, and so there are G12C compounds developed by both Amgen and Mirati that are in clinical trials right now. The mechanisms of action have been worked out for those in several publications. These trials are ongoing, and it sounds like the initial results from the trials are promising. Tumors aren't melting away, but I think they're getting some arrest of tumor growth, and so this is a promising approach to targeting RAS. So when we started this, we realized it's been 40 years. Every pharma that's worth its salt has tried to screen for inhibitors against RAS, and most of them without much success. So I visited Yosemite National Park last year with my family, and one of the really impressive components there is this monolithic rock, which is called El Capitan, which had never been scaled by someone without ropes before. If you ever want to see a film that will inspire you to do things that are impossible, watch this film called Free Solo, because this climber, Alex Honnold, actually spent 3 or 4 years studying the rock face and climbed El Capitan solo without any ropes. And watching it is almost terrifying, but it reminded me of trying to drug a protein that no one's been able to and has no pockets on it. So we'll come back to that analogy. We have a strong structural biology component, so we've been working to determine the structures of all the oncogenic mutants of RAS and see if those reveal pockets that may be targetable. And Sumanshu Dhirendra, who leads our structural biology group, has solved all the oncogenic KRAS mutants, many of these for the first time, bound to a GTP analog. And interestingly, GTP-bound KRAS, or active KRAS, can exist in 2 states, which are referred to as state 1 and state 2. The main difference between those is that switch 1, which is shown in blue here, is out. It's not contacting the protein, or it's a little bit loose. So it's probably not an orientation that's easily bound by effector proteins. So that's considered an inactive state. State 2 is considered the active state, where you can see that the switch 1, the blue, presents a nice flat surface that the RBD interacts with. And this is similar to the structure that Tommy showed you earlier today. But in doing the analysis of these structures, Samanchu was able to show that many of these alleles of KRAS have new pockets. If you look between switch 1 and switch 2 up to the left, adjacent to the GTP, there's a new pocket that opens up in G12C. In G13D, if you rotate it 90 degrees and look down from the top, there's a little cavity that exists there. In Q61, there's also a new pocket that isn't present in the wild-type protein. So this presents the opportunity to potentially target these new pockets in an allele-specific way. So if you found compounds that bound these pockets, they would only go after that allele. They wouldn't touch the other RASs, and those are the type of compounds we'd like to find. This is an example of a collaboration we've done with a group in Israel at the Weizmann Institute near London, and this is exploiting this newly identified pocket. So he's docked in, starting with a library of millions of virtual in silico compounds, is docked in a series of compounds, small fragments, that would fit that pocket, and sends that information to us. We either buy or synthesize those compounds and are evaluating whether or not they're candidates for further development to try to exploit that G12C pocket. In addition, Samanchu has some of these structures are very high resolution, down to 1.1 angstroms, which means you can make very nice crystals, and you can soak in fragments or inhibitor libraries. And at the Diamond Light Source in the UK, they have the capability to soak thousands of these into pre-established crystals. And again, we've done that and are collecting a series of small fragments that may bind to these specific mutants. So this is all work that's ongoing. It's relatively new. We have a lot of small molecules that we believe bind RAS, some in an allele-specific way, and we'll be working to further develop those compounds over the next several years. So again, back to the analogy of trying to climb this monolith that's almost impossible to do. I'm not much of a mountain climber, so I favored this approach. And the idea here is to actually use a rope, and in this case, it's covalent tethering. So this is analogous to what was done with the G12C compounds. And Anna Masiag's group in Frederick is leading this effort. But just very quickly run through the concept of a tethering library. So the idea is that you have small fragments represented here in blue, red, and green that are of different shapes. And if they bind to a pocket on the molecule or your protein of interest, such as KRAS, they'll sit down there. That small interaction is likely to be very transient. It's not stable binding. But if you happen to be adjacent to a cysteine, which has a reactive sulfhydryl group, you can actually form a disulfide bond and stabilize that interaction. So this is a way to probe a protein looking for pockets adjacent to cysteines and identifying fragment binding that you probably wouldn't get out of a screen, but essentially it gives you a toehold to try to at least identify something that might bind weakly and then further develop that compound. And we have two projects or two programs using this approach. One of them is the cysteine at 185. Again, as Tommy described, cysteine at 185 that needs to be prenylated in order for RAS to associate with the membrane, as Tommy described to you this morning. So we did a library screen. Wow. I guess we'll skip that slide. Anyways, we found some small molecules that bound to the RAS molecule, and we could show here using mass spec that these compounds bound specifically to the cysteine at 185. And we can tell that happens because we get a mass addition using native mass spec or intact mass spec of the molecule that we were binding with. This is showing after we've converted the disulfide compound to a covalent warhead. This is the tetrafluoride warhead electrophile, which will actually attack the cysteine and form a covalent bond. In order to use this in cells and to make sure we're actually on target, we developed a pair of cell lines, as Tommy again described this morning, that are based on MEFs, where the only RAS in these cell lines is the RAS that we put back in. So we have a quote-unquote wild-type KRAS that's G12D mutant, one of the oncogenic mutants, that attaches to the membrane through normal prenylation. And then we have another one where we've mutated the 185 to serine so that it can't associate with the membrane, but we've attached it to the membrane through a myristylation moiety that we've added at the other end of the protein. So surprisingly, you can put this myristylated RAS back into cells that have no RAS, and they proliferate and grow. But it gives us a perfect control to ensure ourselves that the compounds that we're working with are on target, that they're hitting RAS. So you can see that when we use this compound in cells, the amount of KRAS goes down. When the cysteine that should be attacked is mutated to serine, the amount of RAS is stable. We don't lose RAS. And also, we can do proliferation assays. So on the orange line, that's the control cell line, and the blue line is the one that we're targeting and preventing RAS from getting to the membrane. I'm not showing it here, but Tommy's done some work to show that these compounds actually delocalize RAS from the membrane. So almost all the RAS you see is in the cytoplasm, not at the membrane. So we believe that these type of compounds are on target and have the potential to inhibit RAS from localizing the membrane and blocking its activity. We wanted to understand how that was working, and we turned to some of our friends who do molecular dynamic simulations. And if you just take the RAS protein with the HVR shown in blue and run a molecular dynamic simulation, it almost invariably, KRAS4B, folds back onto itself, and the HVR looks like it associates with the G protein, potentially forming a pocket between those two helices right there that would have the HVR on top of the pocket. We ran this simulation many times with GTP and GDP bound forms of RAS, and in both forms of RAS, this folds back and potentially folds this pocket, or forms this pocket. In the set of cartoons I'm going to show you right now, it's a representation of multiple molecular dynamic simulations, and the red dot is essentially a click or a picture in time of where the cysteine 185 is. And you can see in the case of KRAS4B, a lot of times it's folding back onto the protein and settling down in between those two helices. In the case of we wanted some controls, so we did the same simulations with the other form of KRAS, an alternatively spliced form, KRAS4A. We don't see the association with the two helices, nor with HRAS or with NRAS, which just looks like it's having a bad hair day. And we can have statistics of these and show that with KRAS4B, most of the time the HVR is settling down in between those two helices, and it's stable there. If you look at the number of simulations where it's stable for longer than 100 nanoseconds, it's much greater than for the other proteins. So we think this identifies a novel pocket that's targetable, and we're working to develop compounds further that bind to that pocket and block processing. We also have generated NMR data, just comparing NMR of the G protein and the G protein plus the HVR, and then we can map the residues on the G protein that change in the presence of the HVR. And again, this is consistent with the HVR folding back and contacting the G domain. So this is potentially a new mechanism, a new pocket for going after RAS itself. So we have these. We're developing some of these in collaboration with a biotech partner, and that program is full steam ahead. During this analysis, we also identified another residue that might be targetable by this cysteine tethering approach, and that's known as H95. When we did the mass spec to see which residues were being bound by the electrophile that we had created, we were surprised to find that on the right, in the lighter color now, you're seeing this cysteine 185, but we are also hitting histidine at 95 in RAS. And that surprised us a bit, but we decided to pursue it further. And if you look at RAS, histidine 95 is actually present adjacent to that same helix where it looks like the HVR is folding back on the protein. What really got us excited is the fact that this histidine is only present in KRAS. If you look at the same position in HRAS and NRAS, they have different amino acids. So this gives you the potential to specifically target KRAS and not the other HRASs. Okay. So we can convert that histidine to a cysteine and then do screens looking for compounds that bind in that pocket. We've done that. So this is the histidine 95 again. This is just a structure showing the G12C compound bound covalently to G12C, but you'll see that it also sits in this same area. And it turns out that some of those G12C compounds are probably also interacting with H95. So this is what we refer to now as the histidine 95 pocket. We have an active drug discovery program going against that pocket. And this program actually looks probably more promising than the cysteine 185 program because we have evidence now that it's also blocking effector interactions with KRAS. So that's a very exciting program and continues again with a biotech partner. If we come back in a year or so, hopefully we can tell you more about that. Okay. We built this disulfide tethering library, or the disulfide library that we used was from an outside source. It was from the chemical discovery program at UCSF. So we decided we wanted a more diverse library, and we've built a new library. We wanted it to be up to several thousand compounds and have good chemical characteristics. This is just a clustering tree showing that the fragments that are in this new library are quite diverse and have the chemical properties that you'd expect or want from a library. Molecular weight's under 300, solubility is good, the right amount of hydrogen donors. You want to stay away from chiral centers, and you don't want too much polarity. And so we've essentially had this library synthesized, and we have it in-house now, and we're starting to use it. At the same time, we decided that this was such a promising approach that we also wanted to essentially do a saturation mutagenesis of RAS and to generate a library of cysteine mutants, protein mutants, that we could use, again, to try to probe the surface and look for small pockets that might be used to identify fragments that bind RAS. And this is just a representation on the left showing an analysis that was a computational prediction of whether or not the side chains of all the surface residues will be exposed or in surface. So all those shown in magenta there or in red above it have been converted to cysteines, and so that's a collection of 95 proteins that we have now. And we know that all the proteins are reactive. We can take a maleimide cross-linker, and again, looking at intact mass spec, you get the shift from the red circle to the right, indicating that all these proteins can be labeled. And so now we're starting a campaign where we'll take our cysteine library of small fragments and combine it with the surface saturation cysteine protein library and do a matrix and try to identify new spots that potentially small fragments would bind to. Of course, if they're positive and we find more small fragments, we'll try to develop those into stronger binders and inhibitors of RAS itself. Okay. The other thing, the second goal was to better understand RAS on the membrane and how RAS interacts with the membrane. And for this, we've turned to people at the Department of Energy Labs. Again, as Tommy had described this morning, it's part of a larger NCI DOE collaboration whose main goal is to use the computational prowess, that is all the big computers that the DOE labs have, to try to advance biomedical research. Some of these are informatic, artificial intelligence, machine learning-based type approaches. The project we're working on is to use hardcore molecular dynamics to better understand RAS biology. So this is just a cartoon of RAS, what it might look like on a membrane bound to RAF, as Sriram mentioned this morning. In this cartoon, which is completely fictional, there's the RAS binding domain in magenta, which binds to RAS. There's a part of RAF called the CRD that is believed to bind to RAS and interact with the membrane. And then, as Sriram pointed out, the kinase domains actually dimerize. So this is a cartoon of one model of what a signaling platform might look like. One of the problems with really trying to study this is Tommy showed you beautiful pictures in vivo of, I call them Tommy's dancing molecules, right, of RAS moving around the membrane. Is that really getting into the molecular details of how this happens in cells is difficult to do, and we just don't have the experimental platforms to reconstitute all of this in vitro. And so we've turned to molecular dynamics to try to solve this. But I just wanted to run through some of the experimental gaps. Even though we have structures of all the RAS molecules, what they look like in the context of a membrane when they're bound to the membrane, we don't know. The dynamics of RAS, how it moves around the membrane. So Tommy's starting to hone in on that and have a better understanding of that. But how it's correlated with specific lipids, again, something we have limited knowledge of. Whether or not there's this long-standing controversy about whether or not RAS multimerizes or dimerizes or aggregates. The distinction between those can be a little subtle, but this idea of whether or not there are actually dimers formed is an open question. And if they do, are there interfaces between the two RAS proteins? Those interfaces could potentially be exploited for drug development. The structure of RAF, Sriram just published a nice paper that partially solves this quandary, but again, that's not in the context of a membrane. That's a solution-based structure. And then actually the mechanism of activation is one of the driving goals of this. So, it's believed that RAF comes to the membrane, interacts with RAS. The order of addition, the activation, the dynamics of all that, the mechanistic details of that are not well known. And then the final one is that what is the lipid environment? What does it really look like where RAS clusters and signals? Okay, so the first part of the story, which we've worked on a lot over the last 2 years with the DOE, is indicated in the box here, and I'll tell you a little bit about that. So traditional molecular dynamics simulations can be done and have been done in atomistic detail. These are done for really, really short time frames because computationally they're very expensive. They take a lot of time and a lot of nodes on a computer to do that. On the left, you have the G protein of RAS and then the HVR shown in green. On the right, it's just two RASs that have been placed next to each other in the membrane, and you're looking down at the top. So those are somewhat informative. They show you that RAS interacts with the membrane, as Tommy indicated earlier today. What we've done is we've combined all of the knowledge and all the experimental details that we've generated over the last couple years, including the dynamics, which Tommy told you about. Jean-Francois has done an extensive analysis of binding of RAS to different liposomes composed of different lipid components. Tommy's group has a lot of advanced fluorescent microscopy that we can use to do analysis and cross-correlations between molecules migrating in surrogate membranes. We've used neutron reflectivity, X-ray scattering to actually understand the protein envelope of RAS and how it sits on a membrane, what that looks like. And then we've also done NMR analysis. And these residues at the top are opposite switch 2 or switch 1, which are the residues that interact with the membrane as identified through NMR analysis. So we've taken all of this information and combined it, and that's been used to parameterize this set of molecular dynamic simulations. So this is the way a physicist looks at the world. So one of the things that's been very interesting about this collaboration, and I would encourage all of you to get to know a physicist someday because they're really smart and they can open your eyes about ways to think about things. They look at a surface, and in this case, it's an undulating surface, and they put a bead on it, right? And that's RAS. Okay, so this is what we call a macro or a super coarse-grained model, right? You just have the surface and this little ball running around on it, and you parameterize it with all the binding affinities and everything that RAS should do, and you just let it run around. They call this a dynamic density force field. Thank you. And then the idea is to merge that or to generate a multi-scale capability where you take it down and actually look at something in coarse grain. And in the coarse grain, you can represent proteins or lipids as a collection of molecules that are defined as small beads that are all pasted together to represent a lipid or RAS. This gives you a little bit more detail, but it doesn't account for every atom. And then you can tie that to what is on the right, it's a charm force field or algorithm for running these simulations where you account for every atom, again, which is computationally expensive. So this is what a biologist or myself would look at this as. in this case, we're looking at a membrane. This is just showing two colors for two different lipids. The RAS molecules are shown as little yellow balls. You can run a simulation. When two of them get close together, as in the green circle, you can then say, okay, let's zoom in on that, and let's start doing a coarse-grained simulation to better understand what's going there. And then you can also extend that and actually do an atomistic simulation to get molecular details about what would happening there. I kind of refer to this as Google Earth for biology, and that's the analogy that works for me. Okay, so we've taken the advantage of the fact that at Lawrence Livermore, they have the second fastest computer in the world. It's called Sierra. It's a new generation of computers. The first fastest is at Oak Ridge, so they go back and forth about who's better. But these have massive computational power, and we were fortunate when these computers were first installed. As they were being validated, we had several weeks where we could run these simulations on these new sets of computers. So the idea was to look at RAS mobility in a more realistic membrane. This has 8 lipids, and start answering some of the biological questions. This is just a cartoon that shows we ran many different simulations. Whenever there was something of interest, that is a RAS in a particular area or two RAS coming together, we looked at a small box. We used a machine learning component to figure out whether or not we'd seen that before. If not, we did a coarse-grained simulation that actually fed back into the larger simulation with new parameters, and this ran as an iterative loop. And we started to get information about what RAS looks like when it comes together and what happens to the lipids. But this is hopefully going to run. Yes? Okay, so this is a simulation where we looked at a micron by micron patch of membrane, which in simulation world is huge. And this has 300 RAS molecules on it. The RAS molecules, you're actually just seeing the shadow of the RAS molecules. And we're looking at the concentration of one of the particular lipids. In this case, it's a charged lipid known as PIP2. And you can see that as RAS moves around this membrane, there's a gradient, which is represented by colors, with the red being the highest concentration of PIP2. And especially when RAS comes close to other RASes, there's an increased concentration of this particular lipid. So again, since we're working with physicists who are very smart and have big computers, we can go back and then do analysis and break this down into the individual grids or patches, as we refer to them. And that was done. And then whenever we found a patch of interest, we ran additional simulations. So in total, the coarse-grained simulation was done on an unprecedented scale, and we generated 116,000 simulations or patches. This is several orders of magnitude more simulation than's ever been done on a membrane with a protein, and in particular RAS. And it gives us enough information that we can start generating statistically relevant observations. So this is just an array showing what happens when you take the lowest 10% of lipid concentration patches and correlate them with the highest 10% of high lipid concentration and you're ranking them based on whether or not you saw a multimer. So essentially it's revealing what lipids are associated with multimers or more than one RAS. And you can see on the second row from the bottom, labeled PAP6, which is PIP2, you get an increase. When you have lots of that around, you get increases in the number of patches that had RAS or multiple RASes in that patch. So it's starting to give us some insights into the interaction between RAS and the lipid membrane, and is leading to a theory where we believe that RAS is actually reorganizing the membrane. It's concentrating lipids out around it. And one of the ways we tested this is to just do analysis, as in the previous slide, and we break it down into two simple scenarios, right? Where you have a RAS monomer or you have a RAS aggregate, two or more together. This was the starting lipid concentration for all the different lipids that were in the simulation. This was the lipid compositions that we ended up with when RAS stayed isolated or in monomeric form, and these are the lipid concentrations when there were multiple RAS in one patch. So you can see that if you have multiple RAS, you actually have less cholesterol. So it selects for less cholesterol. If you have multiple RAS, you start getting a lot more of PIP2. Right? So again, it's showing that when RAS comes together, the lipid environment that it's aggregating in or associating in is quite different from when it's distinct. This is just a model showing, or a movie generated from many images showing this PIP2. Let's see if I can get that to run again. And it shows that it's concentrating PIP2 underneath RAS. As it comes together, there's a concentration of PIP2 at the interface, and then it's redistributed. So again, it's giving us insights that are insightful. And many of the techniques Tommy described this morning we're actually using now to experimentally test these hypotheses. So this computational capability is giving us insights. It's generating hypotheses that we're now testing in the laboratory. And the idea is that this will be an iterative loop, right? If we can generate new parameters that can go back into the simulations and generate new hypotheses to really understand this. So going back to the second objective of better understanding RAS on the membrane, this activation of RAF. Again, we're back to our favorite cartoon. So the second stage of this project will now be to add RAF back into those simulations. So far, it was only RAS and the membrane. But in order to add RAF back in, there's some things we need. RAF structure. Thank you, Sriram is helping us on this and starting to generate some of those structures. Again, we want to understand, what is the order of engagement, and we want to know, again, what that lipid environment looks like that RAF likes to associate with and actually causes signaling to happen. We're doing, many experimental approaches, NMR, there's simulations. We're starting to understand. This is just an example of what the CRD, the cysteine-rich domain of RAF looks like, where it's interacting with the membrane. We have models of what the CRD and the RBD bound to RAS look like. So we're generating starting points for some of the simulations. And this is the paper that Sriram referenced this morning, where he just published, where they have kinase domain dimerization and the way those two domains interact, and also the way one of the auxiliary proteins known as 1433 interacts with RAF. We skipped the one slide. What I just wanted to point out to you is that the RAF has a very complicated activation cycle, and it goes from an auto-inhibited state. And the state that Sriram and John Curian solved is one of the possible many states that are part of the RAF life cycle. So RAF goes from an auto-inhibited state, where essentially it's closed up on itself, and somehow when it binds to RAS or comes to the membrane, magic happens and it starts to unfold. And as it unfolds, different phosphorylations happen, different accessory factors bind or change binding sites, and there's this dimerization of the kinase. So our hope is that by capturing several of these states through structural biology analysis, we'll be able to start feeding that into the simulations and to better understand the mechanism of activation. So again, we're focusing on these two areas. We're trying to target RAS. We're trying to understand RAS in the context of the membrane. We've identified novel compounds that specifically target RAS. We have many screens that are ongoing. I think our feeling is that this is going to take many, many shots on goal in order to find something that's going to work against RAS. We're working with the other labs to do this through computation, and we're partnering. So back to my favorite analogy, right, is that that's the challenge. I think here locally you also have a challenge. And so in order to solve both of those, we have a secret weapon, and his name is Rodrigo, right? We're going to bring him to the RAS initiative. One of the things that many of you have asked me about, just I think because you've heard about it, is we did some crystallization studies of RAS in space. And unfortunately, we weren't able to get significantly better crystals of the HVR coming back. But I just want to close with this slide to suggest that in order to effectively target RAS, we have to be audacious, right? We have to take many chances and try many different ways to do things. And for Rodrigo, if it doesn't work out in Frederick, we can always send you to the space station. So with that, thank you for listening, and I'll be around for the next 2 days, so if you have any questions, I'd love to answer them for you.